thankful to Arima for involving us in this operation. So we're very excited to use HiChIP in our system. This is a HiChIP protocol with antibody against H3K27 acetylated MyoD. And we have applied this HiChIP protocol to our favorite setting. This is the typical myogenic conversion of fibroblast by the ectopic expression of MyoD, the myogenic lineage determination factor. This has been a seminal discovery from Weintraub lab, from Weintraub and Lassar, and has been followed up by illuminating work of Steve Tapscott lab. So why is this a very good system suitable to understand the role of H3K27 acetylation marked chromatin interaction? First of all, because this is a very highly regulated and reproducible experimental system that synchronizes cells at different stage of transdifferentiation from fibroblast to mature differentiated multinucleated myotubes. It also provides one reference transcription factor, which is MyoD, that helps to match HiChIP, IC, and ChIP-seq profiles through the genome and provide a general picture of how the transcription factor that is overexpressed, or actually in this case is epically expressed, these interactions and with respect with the acetylation of chromatin. It also provides a parallel stage-specific gene expression profile, and more importantly, as emphasized by this slide, this system is operated through the ectopic expression of only one transcription factor, as MyoD is unique among all transcription factors for its ability to promote conversion of somatic cell types into skeletal muscles by the ectopic expression without requirement of ectopic expression of other transcription factors. The other good advantage of using MyoD-mediated myogenic conversion is that MyoD is well known as one of the first lineage determination transcription factors that's been associated functionally and physically with the direct the enzymes that are directly implicated in promoting histone acetylation or removing the acetyl radicals from histones. And as a matter of fact, works from Tapscott lab has shown that the ChIP-seq chromatin binding profile of MyoD is highly associated with local histone hyperacetylation. Finally, from a biological standpoint, we know that the expression of MyoD is a fundamental event for the commitment of muscle stem cells into the myogenic lineage. So we know that also in vivo during adult skeletal myogenesis, which is essentially muscle regeneration, MyoD expression turns skeletal muscle stem cells into myogenic committed progenitors and later on multinucleated myofibers. More importantly, we know that epigenetic drugs that impact on histone acetylation like HDAC inhibitors and BET inhibitors can modulate skeletal myogenesis in a stage-specific manner, and we also know that some of these compounds are currently used in clinical trials to promote muscle regeneration. This is our model. As you can see, MyoD is uniformly expressed upon the doxycycline exposure in fibroblast. This is a system where there is a doxycycline regulated expression of MyoD. You see that the large majority of the fibroblasts express MyoD, and at this stage, we have RNA-seq, you can appreciate that at this stage something very interesting happens. And this is the expression of myogenic factors and the consensual erasure or repression of a lot of markers of fibroblastic cell line. And later on, this results in the formation of multinucleated myotubes. So what we have learned using this system is that by combining Hi-C and ChIP-seq, and RNA-seq, of course, is that MyoD operates to promote conversion of fibroblasts into myogenic cells by binding chromatin loops. As Anthony already explained in the previous talk, these loops are organized such that there are two anchors or boundaries which are co-bound by CTCF and cohesion, and that insulate chromatin interactions that refers to promoter and other regulatory elements like enhancers, looping. So we have learned that MyoD can rewire or even change the interactome within these loops by promoting new promoter enhancer interactions. And at the same time, MyoD binding proximity of CTCF at the anchors or boundaries of this loop generate changes in the strength of interaction that regulate the gene expression from the boundaries. So here are two paradigmatic examples. This is a typical genes where MyoD binding that you can spot here at the promoter region of this troponin can instigate a binding which is verified by 3C experiment with a distal enhancer located intragenic in a gene within the same loop where MyoD binds. And here instead is alpha-7 integrin, and you can appreciate that MyoD binding at this time is in a close proximity with CTCF, which reflects the boundaries of this loop, the anchors, and this binding results in the increased trend of interaction between these boundaries and the consensual increase in gene expression of the genes within. There are in the loop defined by these anchors. Still, we realize that the precise relationship between the binding profile of MyoD, its transcriptional activity, and histone acetylation, and the related change of chromatin, three-dimensional chromatin loops was still incomplete, and we decided to address this with HiChIP using the Arima kit. As already shown by Anthony, our samples were within the same range of the other beta tester samples in terms of percentage of captured long-range interactions and the percentage of overlapping between HiChIP reads and ChIP-seq peaks with the same antibody, which was H3 K27 acetylation. Here is the relative enrichment of our HiChIP signal related to the ChIP-seq profile with the same antibody, and you can see that the enrichment shows a very, very good overlap in both fibroblast and MyoD committed fibroblasts into the myogenic lineage that we refer as a myoblast. And there was also a very good replicate overlap ranging around 80% of the common interactions that we capture with the HiChIP in the two replicates, biological replicates of our fibroblast. I should actually point that those are human Imer90 fibroblasts, just to be precise. And the same, we found a consensual 80% reproducibility when we use the HiChIP with MyoD converted fibroblasts into the myogenic lineage. Here is one data that for instance, differs HiChIP with this antibody with the data that we obtained by using HiC and the ChIP-seq, which is immediately portrayed by this graph. As you can see, here we have all the interactions captured by HiChIP that involves promoter, light, and dark blue, other distal non-promoter elements, which are far from the space 1.2 kb and plus 5.5 kb from the transcription site of genes within the human genome. As you can see, there is a partitioning of HiChIP signal in fibroblasts between these two categories of genomic elements. And as you can see, upon the introduction of MyoD, there was a clear restriction, compression of the elements that interacts, which suggests what MyoD indeed does, which is restricting the repertoire of genes that need to be activated. So when we look at the MyoD bound interactions captured with the H3K27 acetyl antibody, we found that most of the interactions that we captured that involved promoters were also bound by MyoD, while only 40% of the interaction captured outside of the promoters were bound by MyoD. Those interactions are likely to be enriched with enhancers, and we conceived that probably super enhancers were important for the myogenic lineage commitment driven by MyoD. So we initially decided to use the computational prediction of super enhancers in our system, which is MyoD mediated commitment of fibroblasts into myoblasts. And we found that 1,343 super enhancers could be actually predicted by ChIP-seq, a profile with H3K27 acetyl antibody. And when we looked at the super enhancers that we could detect with the addition of one of the most important features of super enhancers, which is their ability to interact, 72% of these super enhancers were detected by HiChIP within the other, which are enriched in the enhancers, so it makes absolutely sense. And 80% were detected within the promoter regions. I should say that sometimes, because of the different binning, in the same bin, 5 kb bin, we can find the promoter and part of an enhancer, which is called, therefore, within the promoters. So overall, 90% of the predicted super enhancers could be actually captured by IC, which I believe includes a criteria of specificity provided by the potential interactions between super enhancers. We also looked at the ChIP-seq, HiChIP profiles and the reproducibility of the interaction captured in the fibroblast and the MyoD ectopic expressing fibroblast, which we call myoblast. And you can see that there are pretty reproducible interactions captured in the fibroblast, also in the myoblast. When we look at all the interactions, either induced or downregulated in MyoD expressing fibroblast, we found that those interactions that were increased usually correlate with a better binding of MyoD. So most of the MyoD peaks, by looking at the ChIP-seq profile of MyoD, were found within interaction captured by HiChIP, interaction that were increased in their strand, while those interactions whose strength was decreased were still associated with MyoD binding, but at much less extent. And here's this representation, and you can see here a very interesting feature that could be revealed only by HiChIP. We could not come up with this immediate picture by integrating IC and ChIP-seq profile. You see here related with of MyoD, and that were increased in their strength showed a consensual increase in the H3K27 acetylation. So this clearly associates MyoD binding, increased interaction strengths, and histone acetylation, while a lot of the large majority of interactions whose strength is actually decreased upon ectopic expression of MyoD could still be bound by MyoD, yet the acetylation did not increase. In terms of biological readout, this has a meaning as shown here, because all the increased interactions tend to correlate with the activation of genes implicated in skeletal myogenesis, while most of the interactions whose strength was decreased upon MyoD ectopic expression were associated with the decreased expression of genes that refers to the lineage, fibroblastic lineage. And I should note that most of these pathways are actually anti-myogenic, so it makes absolutely sense that MyoD disarm or downregulates all these pathways or genes networks. And here, I want to show you some examples of how the HiChIP can visualize all the interactions marked by H3K27 acetylation that are instigated by MyoD. So this is the first example. I'll go through a few upregulated and some downregulated genes. So this is an actin that is upregulated by MyoD. You can see that this is the gene, as you can see by RNA-seq upon ectopic expression of MyoD, there is a clear induction of this gene. And you appreciate that in fibroblasts where MyoD is not expressed, there is a clear interaction between these regions, which appears probably close to the promoter, but I don't believe it's part of this promoter, and promotes interactions with different distal regulatory elements that are likely enhancers. Upon expression of MyoD and binding to the promoter of this gene, which is shown by my arrow, you appreciate that now there is a new acetylation that is clearly associated, whose increase is clearly associated with MyoD binding. And these events is sufficient to instigate a huge number of interactions, especially with other MyoD bound distal regulatory elements, which becomes also hyperacetylated. So this is a typical disengagement, simultaneous disengagement of previous interactions, as you can see here, and refocus on MyoD binding chromatin elements that get hyperacetylated and establish promoter enhancer interactions. This is a beautiful example of muscle-specific genes whose expression is a dysferlin, is appreciated by RNA-seq here, is induced by the ectopic expression of MyoD, and this is a very beautiful example of de novo interaction established by MyoD binding at the promoter of this gene with other MyoD bound elements which are likely to be MyoD induced enhancers or MyoD activated enhancers. And this is a paradigmatic example of how HiChIP with H3K27 acetylation antibody can actually reveal interactions between promoter of a MyoD target gene and distal elements that were not captured previously by our combined IC and ChIP-seq analysis. So the interaction that I've shown you in the slides during the presentation was related to this promoter bound MyoD and this intragenic sequence within this gene called LAD1. And that was the only interaction that we captured with our previous analysis, IC-ChIP-seq integration. But as you can see here, with HiChIP, we found a huge number of interactions instigated by MyoD binding to the promoter. And that revealed a huge number of additional putative answers that completely escaped our attention, from our attention in our previous analysis. And finally, I want to show you how this HiChIP kit can reveal a beautiful feature that we're actively investigating, and refers to the MyoD ability to repress gene expression through the disengagement of previous interactions in fibroblasts. You see here a case where MyoD binds the promoter of a gene, which is downregulated. It's kind of paradoxical that a transcription activator downregulates the gene by binding the promoter. But you see here, we are trying to understand why that happened. MyoD binds a promoter that is also bound by CTCF. This could be a boundary of one of these loops. As a matter of fact, the binding of MyoD results in the hypoacetylation of most of the hyperacetylated elements that are supposed to control probably the expression of this gene, and this results in the disengagement of most of these interactions in MyoD expressing cells. This one is even a more drastic phenomenon of disengagement of multiloops that are supposed to drive the expression of a gene that you don't want to see expressed in skeletal muscle cells, because it's GATA6. It's a gene that drives lung differentiation. Those are lung fibroblasts. It makes sense that this gene is expressed in fibroblasts, but as a matter of fact, as soon as MyoD lands on the chromatin, you see that all the MyoD binding correlates with disengagement of a huge number of multiloops, and this promotes the repression of this gene. So with the Arima-HiChIP kit, we found a very easy to use, optimized kit protocol that was working perfectly in our system. It could capture highly reproducible chromatin loops, which are marked by specific histone modification like H3K27 acetylation, and that whose detection typically requires the combined and correlative analysis of HiChIP, IC, and ChIP-seq data. So with the HiChIP, we can reduce the sequencing cost and capture high resolution data. Is a suitable approach to capture chromatin loops between active regulatory elements that drives specific biological programs instigated by transcription factors. And we believe that this is probably one of the best approaches to monitor the effect of certain epigenetic drugs that target the post-translational modifications. In this case, were histone modifications. So it seems to be a suitable system to capture modifications from epigenetic drugs like HDAC inhibitors and BET inhibitors. Let me just thank the three people that have done this analysis. Luca Caputo is the postdoc who made all the HiChIP experiments using the Arima kit. Chiara Nicoletti is a bioinformatic postdoc that analyzed the data, and Alessandra Dalagnese is a former PhD student who is now postdoc at MIT in Rick Young Lab, who initiated the whole project by providing the all IC and ChIP-seq dataset. Thank you guys for your attention.
Great, thanks Lorenzo. Got a couple of questions for you here. I know we're running really low on time. Let's start with maybe the second one, which is, it seems like the next step would be to look at HiChIP using something like MyoD. And do you plan on doing this, and what would you expect, and how would it add to your existing K27 acetylation HiChIP data?
So yeah, I mean, it's clear that the optimal experimental setting would be to match HiChIP profile of histone modifications and HiChIP profile of transcription factors like MyoD. We are clearly working this direction, and I believe that probably even a more interesting setting would be to move into in vivo experiments. So the effort is to reduce the number of cells necessary for this analysis and start to work with FACS isolated cells using two settings of cell types that express and do not express MyoD, which are actually quiescent, for instance, skeletal muscle stem cells, and myogenic progenitors, which are activated upon injury, which is our setting to promote muscle regeneration.
All right, and one other one was, I think it was regarding the evidence that you showed by the gene ontology analyses. Have you done any other orthogonal validation of your loops using non-3C based approaches?
Yeah, can you hear me?
Yes.
Yeah, I mean, this is published in our molecular cell paper that came out in November. So we have used and we continue to use several orthogonal validations. They may range from orthogonal analysis like fish and 3C analysis, but also functional validation with that Cas9. So with CRISPR-Cas9, we could show that most of the MyoD loops that result in the activation or repression of gene expression can be abrogated by eliminating the MyoD chromatin binding with that Cas9. So I think it's absolutely necessary to use this orthogonal validation. But more importantly, when we have done that, that validated our HiChIP data set.
Okay, we have a question also that I think is addressing the ACTN1 gene example, where I guess the question is, there's not a lot of direct interactions in the myoblast at the promoter, but you can see that there's sort of just an increase in interactions just around the gene. Do you think that direct interaction with the promoter is necessary, or just broadly around the gene itself?
We do believe that we have to believe that this is the only perturbation in these cells. The only perturbation is the expression of MyoD and this binding of MyoD. Actually, of course, there is a simultaneous binding of MyoD to these enhancers, which leads to the hyperacetylation and therefore the enhancer activation. So you see that these two binding correlate with a clear increase in interaction between this promoter and these enhancers. Then I would like to drive the attention that these result in the further multi-loops interaction. But I think the first event that triggers the other interactions is this MyoD binding to this promoter. It is also possible that other than a typical MyoD binding, promoter binding, this could also be one of the MyoD binding to a promoter that is also part of a potential boundary, which is CTCF bound anchors of this loop. So this could simultaneously change the interaction strand of these boundaries and generate these loops. We still don't know how all these loops are generated, but clearly they refer to initial binding of MyoD to this element, this promoter, and perhaps the simultaneous binding to other regulatory elements.
All right, and lastly, have you looked at CBP P300 binding and how it correlates with MyoD binding at loci where you lose HiChIP interactions?
No, not yet, but we're running these experiments. We're trying to combine profiles of ChIP-seq of histone acetyltransferases and deacetylases, and then look at the consensual binding with MyoD and loop formed by potential regulatory elements that are enriched with histone acetylation. And of course, we're very interested in testing this setting to understand whether we can generate reliable readouts in terms of chromatin loops after treatment with epigenetic drugs like histone deacetylase inhibitors.
Okay, I think that's all the time we have for questions right now. Joe, I'll hand it over to you to wrap up.